chapter 3 homework, so spend about 5 or so minutes. Anything not clear about chapter 1 and 2, or any homework questions that you couldn't get to?
Each section on the example, people questions.
It'll be roughly. I can't tell you exactly. No, for open notes, right? For the exam, it's open notes. I don't care if you take your notes on your iPad, on your computer, on paper. For the exam, no devices, right? So if you're taking your notes on something, just get them printed out before the exam. All right, no questions about chapter 1 and 2? None? Yeah, that's fine. All right, so let's get to it, right? Okay, now, what we're going to focus on today is binding, right? We're going to look at that through the lens of myoglobin and hemoglobin. Spend a little bit of time on those. Reason we're not covering antibodies and immune systems because we talked about mainly the same things. They just go into detail on how antibodies recognize their antigens, but the mechanism of binding is the same. So we don't have to worry about that. All right, so moving on. So the nature of interaction between a protein and something else depends on the concentrations of both the protein and the ligand, right? So we have a receptor and our ligand. Now, when we talk about two things interacting, often what you see is the partner being called a ligand or a substrate. This is just context specific, right? So typically a ligand is something that binds to a receptor. It isn't necessarily acted on, but does elicit some kind of response. So a cell surface receptor binding, say, estrogen or testosterone, right? That we would call a ligand because it's binding, but that ligand is not being necessarily transformed into something else. Whereas a substrate, we would say something like glucose binding to hexokinase is a substrate. Because though they do still bind and they follow the same mechanism of binding, okay, that glucose is going to be transformed into glucose 6-phosphate. Typically that's the difference here. But the mechanism is the same. So let's consider that we have a receptor and a ligand here, R and L. And these come together, they interact, they bind to form a receptor-ligand complex. And we can have a couple of different types of interactions. You can have very strong binding, right? So it's very tight binding. This means, it's basically as soon as we add ligand, it binds to its receptor, or as soon as we add substrate, it binds to its enzyme, right? So it's very strong. It happens immediately, and happens strong because there's a strong interaction. So we see that as we add more ligand, right, the amount, the percentage of receptor bound to that ligand increases linearly, right? It's just as soon as we add more, right, we bind. So we get a linear relationship here until we get to 100%, right? So what does this mean? Why do we level off at 100%? Right, there's only so many receptors. So what we call this is saturation. You saturated the receptor, you saturated the enzyme. So these types of graphs here, right, we call these a fractional saturation chart, right? We'll go through how to derive an equation to calculate fractional saturation. In other words, what percent of available receptors are bound? And we can also have very weak bindings, so no matter how much L we add, we don't, see a high rise in the amount of receptor in the receptor-ligand complex. Now, in reality, what we see is neither this very tight binding, okay, nor do we see very weak binding. We don't see very weak binding. Why? Why would it be that we don't see weak binding? Now we're in our second week, so we've kind of gotten into groups, and I am going to start calling names off the roster for class participation. Where's Jacob Lodge? All right. So why don't we see this weak binding curve, the blue curve? Yeah, the blue curve. Why don't we see the blue curve in real life?
I don't know.
Don't be scientific about it, just be general. Why would this not be a great curve to avoid?
Because there's no, like, it remains at zero the whole time.
Huh?
It stays at zero the whole time.
Yeah, basically stays at zero, so there's very little interaction. Nothing can happen between a receptor and a ligand if we don't get any interaction. Conversely, right, we get very tight binding, now we have basically no control about how much is binding. As soon as we add a little bit, it's bound, and a little bit more, it's bound. What we'd like to see is something like this, so we get a relationship, right, a slower relationship between how much ligand there is present and how much is bound to the receptor. And we can quantify, right, the extent to which it's bound, right, with numbers. So here, what we see is L half, sometimes referred to as L50. Now, what this is showing us is, what is the concentration of ligand necessary such that half of the receptors are occupied, right? So notice here we're at 50% or 0.5. Follow that over. This ligand concentration we define as L1 half. Now, in reality, okay, what we see, what we observe is that we purify an enzyme or a receptor and we analyze its L50 or its L half. That L half is usually in the range of the physiological concentration of that ligand. So for example, if we have a receptor that binds estradiol, let's say we'll see an example of that in just a minute. We have a receptor that binds estradiol, the L50 for the estradiol receptor to estradiol is about 1 nanomolar, right? The concentration that we see of estradiol in the body is, you know, right around 1 nanomolar, give or take, depending on the time of the day, right? Why is that, right? Why might we see an L50 that vaguely corresponds to the physiological concentration of that ligand? All proteins, all receptors are going to have their own specific L50 or their specific ligands. Chris Kim, where's Chris Kim? Why is L50 or L half, why is that usually right around the physiological concentration that we find the ligand at in the body?
Because I'm not too sure.
All right, that's all right. Take a guess. What would happen if the concentration were the concentration of, say, in this case, estradiol? What if it were less than L50?
Would have different reaction.
Huh?
There'd be different like result.
Different result, what would be the result? So here we're talking about binding, so look over here, if we're at less than L50, what would happen to the percentage of receptors bound?
They'd be less bound.
It would be less, and if it's more than L50, it would be more bound, right? So enzymes, receptors, have evolved to have an L50 that allows them to respond to changes, to physiologically relevant changes in their ligand concentration. So here, for example, we don't want to trigger the estradiol receptor if the concentration of estradiol is low, right? So it's got an L50 of 1 nanomolar, right? So if we're at, you know, early in the morning, let's say, we might have 0.1 nanomolar estradiol, right? Some of us. So we'd be down here, about maybe 5 or 10% occupied. This would not be enough to trigger the response. Whereas later on, we might be at 1.1 or 1.2 nanomolar, and now we're above 50, and we are able to trigger the response. And the same is true for any protein or receptor interaction, right? Now, remember from chapter 1, the shape determines everything about chemistry. In this case, the shape determines the specificity and the strength of binding. Let's again go back to the example of the estrogen receptor. Recognize this estradiol. Now, if we look at estradiol, right, here's the structure of estradiol, here's the three-dimensional representation, both from the top and from the side. We look at testosterone, right, a very similar hormone. It's got almost identical structure, okay? It's got 4 rings, it's got a hydroxyl here, methyl here, no methyl down here, but importantly, the A ring in estradiol is aromatic, planar, good, but not in testosterone. So if we look at it from the side, right, well now testosterone's got a little bit of a kink in it, effectively making it thicker, okay? Additionally, this has different sites of interaction. Here we have, what, this double bond O here, what could this be? what kind of interaction would it take place? A hydrogen bond. Is this a hydrogen bond acceptor or donor? Acceptor. What about over here, estradiol? Donor, right? So these won't be able to make the same types of interactions, all right? This won't be able to fit what it does. So how do receptors, how do proteins distinguish between their different substrates and ligands? Well, that has to do with the concept we call the active site. Right? So remember from chapter 2, a protein adopts its primary, well, adopts its secondary and tertiary structure. Sometimes it's an ordinary structure, but here just the tertiary structure. So all the alpha helices and the strands, the sheets and the loops and the turns all come together in a specific way. And now we have a binding site for a receptor or an active site for an enzyme. And lighting, right, so here's where estradiol binds, or binds. We determine this using crystallographic or NMR methods, or cryo-EM. If we have time, we'll get to that at the end of the semester. But we know the structure of the receptor, we know the structure of estradiol, we know the structure of the complex together. What we see is that there's a discrete pocket in which it binds. And this is true of any enzyme or receptor with its ligand or substrate. Lining this pocket are, of course, amino acid residues. The entire protein is made up of amino acid residues. The greater function of the rest of the protein, not the active site, okay, is to make sure that the active site has both the correct composition of amino acids and that they're oriented in the correct way, so that they can make interactions with their substrate, that those interactions are maximized when it's the correct substrate, right? So if we look at, for example, here, how does the receptor differentiate between estradiol and testosterone? Well, first, testosterone is slightly thicker, right? So it's going to have a harder time physically fitting into the active site, into the binding pocket, right? So that's its first strike against it. Second, all right, testosterone has slightly different functional groups there, so it can't make as many favorable interactions with the estradiol receptor active site, right? So we take a measurement of the L50 for estradiol is about 1 nanomolar, right? So the L50 for estradiol with the estradiol receptor is about 1 nanomolar, whereas for testosterone is about 1 millimolar, so 1,000 nanomolar. Okay, even these very small changes in size and in the composition of the functional groups here, they are enough, right? They are enough to make testosterone by 1,000 times weaker. Right? If we think about it, if the L50 is 1,000 nanomolar, that means we need 1,000 nanomolar to get 50% occupancy, whereas we only need 1 nanomolar of estradiol to get 50% occupancy. So that's a stronger interaction, again, due to the correct size and being able to form better interactions. All right, now we can quantify how much receptor is bound, okay? And this is called, again, fractional saturation. In other words, what percent of the total amount of enzyme we have that is in a receptor ligand complex or an enzyme substrate complex. We'll see this again when we talk about enzyme substrate complex and enzymology. All right? Okay, so for this process here, R plus L yields RL, right? The receptor plus the ligand yields the complex. This, of course, is just a process, and like any process, it reaches an equilibrium. And in this case, we call that equilibrium Ka, not to be confused with the acid dissociation constant, Ka. This is identical in how it looks, but it's very different in what it means. The association constant describes this equilibrium between a receptor and its ligand. Of course, just RL divided by R times L. Now, if we take the reverse of this reaction, in other words, the complex yields free receptor and free ligand, well, now that's a dissociation reaction. And we can just say that Kd, the dissociation constant, is equal to R times L over RL, which of course is just 1 over Ka. Right? So Ka is the association constant. Kd is 1 over Ka. And by far the more useful term here is Kd. Kd, as we'll see, is going to give us an indication of the strength of binding between a receptor and its ligand or an enzyme and its substrate. The lower the Kd, the stronger the binding. You'll see why that is in just a second. Right, now fractional saturation, effectively, how much R is bound, we call that Y, all right? And we can say that just in plain terms is the number of sites occupied divided by the total number of sites, right? So how many sites are occupied is just RL, right? RL is the number of sites occupied, right, as a concentration, but we can just convert that to mole, right? So now we have a number. Either way, RL divided by total number of sites, which of course must include RL, plus free receptors, right? These are not part of a complex, but they are sites, so they're part of the total sites. So we have RL divided by RL plus R. This is fractional saturation. Now, we don't know RL, right? We don't know right now how many specific receptors are part of a complex, but we can deduce it using this association constant. You can say that RL just equals Ka times R times L. So we can sub that in here and here to get this expression, KARL divided by KARL plus R. Now we do a little bit of fancy math. We factor out Ka times R on the top and bottom. On the top is easy. On the left term here is easy, but now we have to multiply by 1 over Ka. Right? If you take your time, you'll see that Ka times R times 1 over Ka is just R, like we started with. So this all simplifies down to L over L plus 1 over Ka. Of course, we know that 1 over Ka is Kd. So we can simplify this further to say that the fractional saturation equals L over L plus Kd, right? All right. Now, what does it mean then when the concentration of L equals Kd? What does that mean for the fractional saturation? If L equals Kd, Rishikesh, I met you before. Yes.
That means 50% of the sites are occupied.
Explain why.
But if L equals KD, let's just say L is 0.5, KD is 0.5, then it'd be 0.5 over 1, and then because of the Y percent occupied for the sites, that would mean that 50% sites occupied.
All right, very good. So we say that L equals Kd, though what this simplifies down to is L divided by L plus L. So L over 2L, cancel the L down, we're left with 1/2. Kd tells us, just like L50, the concentration of L required to reach 50% occupancy. So Kd and L50 are functionally the same thing. So this could be rewritten also as L over L plus L50. Right? We're going to see that when we examine hemoglobin in just a second. Right? So again, L equals Kd, Y is 0.5 to 50%. Kd is an indication of the concentration at which the receptor or protein goes from mostly free to mostly bound. Okay? Which again is why we see that the L50 is typically in the range of the physiological concentration that we find the ligand at. Okay? Alright. Now to put this into perspective, let's examine myoglobin first. So myoglobin binds oxygen as its ligand. Okay, and what does this mean? Well, that means that we can put together a fractional saturation curve, which when we're talking about myoglobin and hemoglobin, we refer to as a loading curve. Right? So we say Y here. PO2, this is what we're going to use in lieu of ligand concentration. Of course, O2 is a gas, so and we're breathing oxygen, not inhaling or not sucking it in. So instead of using molarity here, we're going to use torr, right? So it's the portion pressure of oxygen in torr, and what we see is a hyperbolic loading curve like that. Okay? So it exists or exhibits a binding just that we derived in the last 2 slides. Torr, by the way, is the same as saying millimeters of mercury, right? So if you're going into the medical field, millimeters of mercury. Anyway, right? The P50 for myoglobin is 2 torr. Okay? Above 2 torr, more than 50% of the myoglobin is bound to O2. And below 2 torr, myoglobin is mostly free. Okay? Now, here, right, is an actual graph, right? So just plug this into Desmos, and we say the fractional saturation Y equals PO2 divided by PO2, so L over L plus KD, or L50 in this case, 2 torr. Right? Well, now we see at 2 torr, we come up, we're at 50% saturation. Here's that blue line, right? And we get this hyperbolic curve just like we described. In fact, any enzyme that follows this type of binding, okay, will exhibit a hyperbolic binding curve, or in this case, a loading curve. Now, myoglobin is going to be an exceptionally bad oxygen delivery system, right? So examine this here, and I'll tell you that the partial pressure of oxygen in the lungs is about 100 torr, and the partial pressure of oxygen in the tissues is about 40 torr, right? So at 100 torr, what is myoglobin really good at doing in terms of oxygen? Storing it, binding, right? So if we look at 100 torr, we see that we're at basically a 99.9% binding. We'd never be at 100, of course, right? Because if you look over here, L over L plus KD, the only way that we can be at 1 over 1 is if KD is 0, and KD cannot be 0, because that would mean infinite binding. So KD cannot be 0, right? So this is not great. All right, I'll just stay over here. Good. Now, in the lungs, of course, it's going to bind. But what is myoglobin really bad at doing with oxygen? Letting it go, right? So if we come down here to 40 torr, we're still at about 95%, right? So we're not really delivering that much oxygen, which is not good, right? The oxygen is going to make none of myoglobin, and it's not going to get to the tissues. For that, we need something else, right? But before we get to something else, let's examine how it is that myoglobin binds oxygen, because hemoglobin, when we get there, is going to bind oxygen in a functionally identical way. Right? So myoglobin has something we call a prosthetic group. And that prosthetic group in this case is heme. And a prosthetic group is exactly what it sounds like. It's a group that's prosthetic to the receptor or enzyme. That means that it's necessary for that particular enzyme to function. In this case, myoglobin nor hemoglobin would be able to bind oxygen without this heme prosthetic group. So you see the structure of heme at the bottom here. Heme is composed of a protoporphyrin, which is a tetrapyrrole here, and in the ring you're not going to need to memorize the structure, don't worry. But it's got an iron in the center, right? And it's only when the iron is in the center that this becomes specifically a heme, right? Now, this iron has 6 electrons that it can share, right? So it's got 6 coordinate covalent sites. 4 of those sites are occupied by the nitrogens on each of these pyrrole rings in the protoporphyrin. So holds that iron there. That leaves us with 2, right? We have 1 coordinate covalent site going away from you, and 1 coordinate covalent site going directly towards you. All right? So what does this heme do? Specifically, what does this iron do? Well, this iron is typically found in the ferrous state, Fe2+. Right? That's the way we normally find hemoglobin. And in the Fe2+ state, it's able to bind oxygen. Right? So before it binds oxygen, we see that it's always bound at the 5th coordinate covalent site with the proximal histidine. Right? This proximal histidine, we go back to the structure of myoglobin. Right? Here we have our heme. Here we see that it's surrounded by these helices. On this helix here, right? At this helix here, there is the proximal histidine. Remember, the functional group on and off the helix sticks out away from the axis of the helix. So we have the histidine sticking out that way towards the heme, and that's what we see here, right? This proximal histidine here on the helix is sticking out at the 5th coordinate covalent site, right? So that's always there. Now, with myoglobin, this tends to actually not matter at all, right? But when we get to hemoglobin, this proximal histidine is going to become very important. Now, once oxygen binds to this Fe2+, right, you see something interesting happen. Here, the Fe2+ is slightly too large to fit inside this pore of the tetrahydrole, right? So it sticks out and puckers that heme. Right? When oxygen binds, now this is converted to the ferric Fe3+ state, right? And now it's got a shorter effective van der Waals radii. It can unpucker because it fits inside that pore, right? So now we have the iron in its 2+ state, all 6 coordinate covalent sites being occupied, 4 with the tetrapyrrole, 1 with the proximal histidine, and 6 with the O2, right? This here is important, both for the case study and for life in general, all right? Here what we see is that though oxygen binds to the Fe2+ state, once it binds, right, really what we have, we can consider to be a superoxide O2- with Fe3+, right? Why is this important? Well, O2- is a type of reactive oxygen species, right? If that were to be released, well, now that's detrimental to the cell, right? That can go off and interact with DNA and RNA and proteins and destroy them. Additionally, right, what would be to happen if this O2- were to dissociate at this point? What would we be left with? Because of course, O2- there. Huh? ferric hemoglobin, right? And what do we call ferric hemoglobin? Huh? Oh, sure, methhemoglobin or methemoglobin. Forgot we're talking about myoglobin. Yes. So methemoglobin, and methemoglobin is unable to bind oxygen. So that's a dead end. All right. Now there do exist mechanisms to reverse, right, to reduce that ferric Fe3 plus into ferrous Fe2 plus, right, which was part of that case study. But in individuals deficient in cytochrome B5 reductase, right, that's not possible. So this myoglobin, hemoglobin that builds up remains. Okay? So how do we prevent then the release of this O2 minus? Well, we can't entirely, but we can help to prevent it. And that's done by the distal histidine, quite separate from the proximal histidine, simply on the other side of the heme. So if we come back up here, the proximal histidine is here at the fifth coordinate covalent site. This finds oxygen on its opposite face. And then over here on this helix would be the distal histidine. And what the distal histidine does is to provide a type of weak interaction with the O2. So what type of weak interaction is it providing? It's just an H bond, exactly, right? Here in this case, the histidine is providing a hydrogen bond donor, right, to the O2. So stabilizes this O2. Because now in order for the O2 minus to leave, all right, we would have to break the hydrogen bond. So this is unfavorable. That doesn't mean that it never happens, it does happen, but it happens now less frequently. Now, take a second and examine this, right? This incorporates things that we've learned in chapter 1 and in chapter 2, things that you should have when you go off the top of your head. There is some kind of problem with this, and it has to do with pH. Look at this drawing here. And it doesn't seem to make sense. What's the pK of this? This one's easy. This one you should know off the top of your head. 6, right? That's the pK of the side chain of this proton here. At what pH would we hope to find most of our blood? 7.4 thereabouts. So is this histidine going to be protonated or deprotonated? Is the pH higher or lower than pK? Is 7.4 higher or lower than 6? There we go. So is it going to be protonated or deprotonated? Deprotonated, exactly. And yet, we have this hydrogen here. If we didn't have this hydrogen here, we wouldn't be able to form this hydrogen bond. The pKa values that we see for these residues, these amino acid residues, are true. If you look at that table, it is 6. If you have a pure mixture of histidine, you measure it, the pK is 6. But pKa values can change depending on the electronic environment in which that amino acid residue finds itself. In this case, this histidine is inside of myoglobin, all right? And the pKa value has changed, right, to slightly more than 7.4, 7.5 or so, right? This is possible. In this case, because now the apparent pKa is higher than we would normally expect, now this remains protonated at 7.4, we can stabilize the O2 being bound by formation of this hydrogen bond. So the pK of histidine I drilled into, right, should be 6.0, which means that pH 7.4, that proton over here should be gone, right? But it's not. Because the pKa of an ionizable proton can change, right? And it can change based on the electronic environment that we find that residue in, we find that proton in. We're going to do some examples hopefully later on involving exactly this. So in this case, this histidine, this proton on the histidine, doesn't have a pKa of 6.0. It's got a pKa closer to something like 7.5, right? So that now the pH is lower than the effective pKa, so it remains remains protated, and it can donate this hydrogen bond to the O2. If that weren't the case and the pK were 6, this couldn't happen because that proton would be gone, right? And now hemoglobin is much better oxygen carrier than myoglobin. Myoglobin we find where? Where do we find myoglobin? In the muscles, right? So, I don't know, freak anyone out if you're a vegetarian, right? You're eating your steak and you cut into it and all that. That's not, blood necessarily. There's very little hemoglobin there. It's all myoglobin, right? We find that in our tissues. Why? Because our tissues, or the muscles specifically, require a lot of oxygen. We cannot generate enough oxygen or energy to function without oxygen. We can generate energy without oxygen, but not enough to live, right? So this makes sense, right? So when you want myoglobin, that good electron stealer, right, in the muscles, so it can steal it away from wherever it's coming from. Where it's coming from is hemoglobin, right? We call hemoglobin HbA for adult hemoglobin. This differentiates from HbF fetal hemoglobin, or HbS for sickle cell hemoglobin. So HbA is a much better carrier of oxygen, specifically because it's worse at binding oxygen, right? So if we look at the loading curves for both myoglobin and hemoglobin, we notice a couple of things. First, right, what's the first thing you notice about this chart, comparing hemoglobin to myoglobin? Huh? The curve is sigmoidal, right? So instead of being hyperbolic, we just jump up, right? We start off slow and then ramp up. This is hallmark of enzymes that are either cooperative or allosterically regulated, or both, right? Like hemoglobin is both allosterically regulated and cooperative. What else do we notice about the curve?
It's higher.
P50 is higher, which means
That binding as good.
the binding to oxygen isn't as good, right? And this is on purpose, right? Here we go. Now, if we look at the structure of hemoglobin, right, let's take a second here. We see that hemoglobin is not like myoglobin, which is just one polypeptide chain. Right? So we go back and look at the structure of hemoglobin. That's it. Okay, it's a very small protein. Hemoglobin is comparatively large, about 4 times larger, because it's made up of 4 different polypeptide chains. It's got a defined quaternary structure. None of the chains on their own are able to do anything useful. Okay, it's made up of 2 alpha chains and 2 beta chains. And it's going to be useful in just a second to consider hemoglobin to be a dimer made out of dimers, right? So we have an alpha beta dimer with an alpha beta dimer that fit together, right? The reason is that these 2 alpha beta dimers are going to twist against each other when it binds oxygen. We'll see why and how that happens in just a second. Right, so we have 2 alpha chains and 2 beta chains. Each chain is contains a heme prosthetic group, and each one can bind its own O2, so we get a hemoglobin that can bind 4 O2s at the same time, right? But it doesn't bind them all the same, and that's the cooperative nature of hemoglobin. Right, so again, the HbA is a tetramer. It's got a P50 of 26 torrs. Of course, somebody said over here the P50 is higher, okay, so it's worse at binding oxygen, and it's got a sigmoidal loading curve, somebody else said, due to its cooperative nature. Okay, and both of these put together, the higher P50 and the sigmoidal loading curve, is going to contribute to why it's a good oxygen carrier, not necessarily a good oxygen binder. Right? So why do we want to use a more complex tetramer instead of a monomer, such as myoglobin? Right? So the first is that multiple subunits facilitate this phenomenon of cooperativity. Monomeric enzymes or monomeric receptors like myoglobin cannot exhibit cooperativity. What do we mean by cooperativity? What does it mean to be a cooperative enzyme? Sure, when one binds, it increases the likelihood of the other one binding. That is true for hemoglobin more generally, right? What is a cooperative enzyme? That is correct, right?
I guess generally it would just be something that works together.
Exactly, something that cooperates. It works together. And in this context, for hemoglobin, right, the first oxygen to bind is difficult, right? But that makes the next oxygen to bind a little bit easier. And the third one to bind even easier, and the fourth one to bind easiest. Okay? So multiple subunits facilitate this cooperativity. Why do we care about cooperativity? right? Well, let's remember cooperativity is what gives the curve that sigmoidal shape. So what if we were to design, right, a hypothetical monomeric oxygen carrier, okay, that delivered as much oxygen as possible? So, with a bit of fancy math that I don't quite understand, the best possible situation we can have with no cooperativity is to deliver 38% of the total oxygen that binds. If we look at 100 torr versus 20 torr, right? We can deliver 38. But if we have a cooperative enzyme like hemoglobin, now we can deliver 66%, right? So 38% is not really viable. Okay, we need more oxygen than that, and specifically, we need different oxygen amounts in different tissues, right? We can have an exercising muscle, which has a PO2 of about 20 torr because it's using a lot of oxygen, so the oxygen concentration is low. Or we can have a resting muscle where the PO2 is about 40 torr, since it's not doing as much work, right? If we look at here, at rest, at 40 torr, we're only delivering 21% of the oxygen that hemoglobin bound, but at 20 torr, we're able to deliver 66% of the oxygen bound, right? So this sigmoidal loading curve, in addition to being able to deliver more oxygen, allows hemoglobin to respond to the different needs of the cell just based on partial pressure, right? So no extra regulation is necessary. This is just a fact of the enzyme. Additionally, if we didn't have cooperativity, well, here we're at 20 torr, delivering 38%, but if we go over here to 40 torr, right? Looks like now we're delivering about 25 to 30%. Okay, so not as big of a difference between that and 21 to 45, 21 to 66%, right? So there's less of a response to physiological conditions. So the loading curve, the sigmoidal loading curve, allows acute response to physiological conditions. So how's this cooperativity achieved, right? So we have fully deoxygenated hemoglobin in the 10 state, okay, in the T state, or we have fully oxygenated hemoglobin in the R state. Yeah,
why do we want hemoglobin to be binded?
say that again?
Why do we want the hemoglobin to bind to the oxygen if it's supposed to release it?
Well, it has to bind it in the lungs, because that's where we take in oxygen, and then it has to release it once it gets to the tissues. Right, so it's got to both be good at binding oxygen and be good at releasing oxygen. Myoglobin is very good at binding oxygen, but very bad at releasing oxygen. But we need an oxygen carrier to do both, because we can't circulate enough oxygen just dissolved in fluid to meet the needs of the body. So we need a way to carry that, right? And the way that we carry that is on a protein, in this case hemoglobin. So we want it to be good at binding oxygen when it's supposed to be binding oxygen, i.e. the lungs, and be good at releasing oxygen when it's supposed to release the oxygen in the tissues. So we need to be both. Hemoglobin is only very good at one, or myoglobin. Hemoglobin is good at both. Right? So in the T state, okay, we have fully deoxygenated hemoglobin. We have fully oxygenated hemoglobin in the R state. So that's the left, the right, on the top here. Okay. If we look at the binding curve for T state hemoglobin, right, what we see is very similar to what we saw at the beginning of class. Very weak binding. Okay, this makes sense since T state hemoglobin is deoxyhemoglobin, and because we know it's cooperative, the first oxygen combined is going to be very difficult. So if we have fully T-state hemoglobin, it's not going to bind oxygen very well. It's going to be very good at releasing oxygen. Right? So when we want to release oxygen, what we want to do is stabilize the T-state. Conversely, we look at the R-state, which is very good at binding oxygen. Well, now we have a curve that looks functionally like myoglobin. Okay, so it's very good at binding oxygen, but like myoglobin, it's very bad at releasing oxygen. So we need hemoglobin to exist in this in-between state, right? The observed hemoglobin binding curve. It can neither be R, right? Because then we wouldn't deliver enough oxygen, nor can it be T, because then we wouldn't bind enough oxygen, right? So we need to switch between R and T depending on how much oxygen there is, okay? Now switching from the T to the R state rotates the alpha-beta dimer relative to the other alpha-beta dimer about 15 degrees counterclockwise, right? And when this happens, right, notice that this pocket here, this little pore, closes up, right? This is going to be important for regulating how much oxygen hemoglobin can deliver. right? So switching from the T state to the R state requires this twist between the alpha beta dimers, and that closes the pore there, okay? Now, how does hemoglobin go from R to T? Well, here we have two models, the concerted model and the sequential model, and neither is entirely correct, right? Neither is entirely right. What we have is the concerted model, which explains oxygen binding best at high occupancy, so when there's a lot of oxygen present. In the sequential model, it explains it better at low occupancy. In other words, here, right, in the concerted model, what we have is hemoglobin existing either totally in the R state or totally in the T state, right? And we have an equilibrium going from hemoglobin binding one or no oxygen to binding one oxygen. Now here, in this case, we start here. We see that we have mostly the T state, right? The T state is strongly favored because we have no oxygen. We have 99% this and say 1% this. And this has a very low forward rate to go bind one oxygen, but this has a high rate to go bind oxygen because it's in the R state, right? So this one goes forward quickly, this one really doesn't go forward. And there's very little of this, so overall going from this state to this state takes a long time, right? Because we have so little of this to go over here, and this is not going to go over here. Now if we look over here, right, once we've found the first oxygen, notice the equilibrium between these two states now still favors the T state, but not as much as it did over here. Right, so we have more R, which means that we can go from this state to this state a little bit faster. Right? Now we're at roughly equilibrium, right? So we have about equal amount R and T, which means that this can go over here more quickly, right, than it did before, and so on and so on. Right? Down here, the sequential model doesn't describe a hemoglobin that exists either completely in the R state or the T state, but instead can have individual subunits or individual chains that are in either R, T, or a superposition of both. Right? So if we start totally the T, right, this K1 going from this state to this state is going to take a long time because T, the oxyhemoglobin, is not going to want to bind oxygen, but it will. Right? There is still some forward rate, even though it's very slow. But now this converts whatever subunit bound to oxygen from the T state to the R state. Now it's binding oxygen. And when this happens, this communicates to the other subunits nearby, right? The ones adjacent to it. Hey, I found an oxygen, so that makes it easier for those to bind an oxygen. So we're in kind of R/T state here, right? So now either one of these can bind an oxygen easier than this here. So that's what we see here. Could have been this one or could have been this one. This cartoon is this one. Now both of these are in the R state. They're both making contact with unbound subunits, converting them into the R/T state. Which means either one of these can bind, right? And then now the next one can bind. The reason why we say that this one explains it best at high occupancies is because this makes sense, right? But going from this state to this state, right, doesn't really make a whole lot of sense here because since we're at 99.9% T, going from here to here seems like it would just take entirely too long, right? But once we've got oxygen bound, this makes sense. Over here, this makes sense in the beginning, right? This is slow, but will happen. Once we reach over here, right? Why do we say that having two of these in the R/T state? Why is this better than having only two this way in the R/T state? So this is less useful at high concentration of two, and this is less useful at low concentration of two. But either way, there is some communication between the subunits, so that the first oxygen makes binding of oxygen at the other subunits slightly easier. How is this communication achieved? Well, this goes back to that proximal histidine that I told you didn't matter for my hemoglobin. Right? In this case, the proximal histidine does matter quite a bit for hemoglobin. Right? So remember what happens when O2 binds to the 6-coordinate covalent site on that iron. Right? Remember, it shrinks, it depuckers, and it pulls up on this histidine. Right? So that iron now shifts up into the plane of the heme, and because it's in a hydrogen bond with that or in a coordinate covalent bond, with the proximal histidine, it drags the proximal histidine up with it. And of course, the proximal histidine is part of the alpha chain or the beta chain, what have you. All right. Here, it's going to pull up on the helix that the proximal histidine is a part of. Right. When it pulls up on this helix, it moves this beta loop over here, and this beta loop is right at the interface between the two alpha beta dimers. And that movement in that loop connected to this F helix can be sensed by the other alpha beta dimer, and it makes it easier, it changes the conformation of that alpha beta dimer so that it can bind oxygen more easily. Right? So it's the movement of this F helix, right, or the histidine connected to the F helix that communicates changes to adjacent subunits. This has been proven by experiments. This is in a model. This is actually what happens. Right? So if you get rid of this proximal histidine, all right, what would you expect the loading curve of hemoglobin to look like? Linear? No, not quite linear. Would it be cooperative any longer? Would you be able